Uh,
Welcome back.
yeah, so we'll come back after our hiatus, I guess. Yeah. Um,
Or Ruby balked around the world for too long.
yeah, yeah.
Didn't have time to stream.
But you said that you want wait, but not for too long because you said that you wanted to go continue vacationing for like a month, right?
That's true. I would totally have gone longer if only I had the time and the money. It would have been nice. It would have been nice to like camped in Iceland properly and just spent more than 3 days in a city.
That would be
For
nice. Yeah.
yeah, it was very
Camping in
nice.
Iceland sounds really cool. But
Yeah, I think you have to like find the window where the weather is nice. And I was very lucky and got basically perfect weather for like an entire week. Um
Mhm.
and now it's like really cold. I did miss all the auroras. I have to say like this is a really good time to get back into streaming because we can like do an update on all the like cool things that are happening in the sky. So like September had some of the biggest solar storms, and so when there are solar storms, our magnetic field kind of goes wonky, and when our magnetic field goes wonky, we get auroras, and it was like apparently like some of the most beautiful auroras we've seen in a while. Missed it all because it was no longer in Iceland.
Yeah.
But I think it's some of it's still going on now, and then we have some like really cool stuff coming up like
There's gonna
next
be an
week.
eclipse, right?
Yeah, there's gonna be an eclipse next weekend on the 14th. There's also the moon is gonna just be like right next to Venus, so you can have like the two brightest things like side by side fairly soon. Yeah, it was just like it was kind of an exciting time to be like, hey, like if it's not cloudy, go look outside at night. Or if you can't sleep, go look outside.
Go outside.
Yeah. Well, also the eclipse, like I figured, it would be fun to build a pinhole camera at some point,
Mhm.
before then. Maybe we can do it next stream because we still have time.
Oh yeah, do a
Uh,
stream where we just build a pinhole camera.
yeah, with like a Pringles can or like a cardboard
That'd be fun.
box,
Yeah.
right?
Yeah.
Yeah, and then like we can just talk about like we can chat about like why and like what happens and
Eclipses and like how the optics work, and yeah, that would be fun.
yeah. Oh, and we can do like a history of like what other humans have thought about the eclipse,
Mhm.
or like what
Which is
happens.
really
Like
quite strange.
yeah, like humans have thought like very varying things, like from good omen to really bad omen to like like sacrifice your firstborn and to like let's plant the trees. Like they've done
It's
all
kind of
sorts of
interesting
things.
because so the last time there was an eclipse was like a total solar eclipse. One of the things I remember reading about and then noticing is that it like really freaks out the birds and they just start like squawking and flying everywhere. But like in a way, it also does that to humans.
Yeah, well, like we are also squawking and running
Yes,
around.
we're also just like it got slightly darker when it wasn't supposed.
Yeah, that's great. Are I don't think I'm in the path of a full eclipse, so I might actually only get to see a partial one. In which case, I really do need to build a pinhole camera,
Yeah, I think
which
we'll only get the partial.
we'll get the partial. I remember people like driving to see it because it was gonna cut through the states. People like drove like west to see it from here, which is
Mhm.
cool. Um, yeah, I think
Well,
that would be
also unrelated, you'll notice that Nadira is not here, but she's still like here in a general sense. Like she's not dead or anything. Just like she is writing some
two
papers and
papers
it's very busy.
simultaneously.
Yeah.
So we'll
So
get her.
one of the reasons that we have a hiatus is that like both Ruby and Dira are moving to their new labs. So it's a very exciting but busy period of time for us.
And we're starting new things, which is exciting.
Yeah.
Um, yeah, but we thought this week I was like, wait, they just announced the Nobel prizes for physiology and medicine, and also chemistry and physics, and like we should just recap it because I feel like this is for the lay audience, and I like this is the biggest, I guess, like the Oscars or the Academy Awards. You know what I mean? Like this is the equivalent in the science world.
Mhm.
Like let's you know like let's
Like
talk
talk
about it.
about yeah.
Yeah, let's jump on the bandwagon and talk about it. I'm actually really happy with this year's this year's like awardees. Like I'm not in this field. I just like we just did some aggressive reading for the past
Yeah,
day,
yeah.
and it was like, all right, now we want to know about the things. Um, what did you know about these scientists before today?
Like really nothing. Like
Okay, great.
I
Yeah, me
hardly
neither.
heard of them at all. Yeah.
Yeah, no, me neither. Which is, I think that's like one of the coolest things about like these high profile prizes is that it kind of like elevates something that we know is really important. We just didn't have a name to go with it, and now everybody's talking about it. At least for like a couple weeks, like these are the topics that people are talking about. And it's like, oh
Yeah.
yeah, that was really really important. Do you want me to go first? Because
And
I
yeah, I was gonna say I feel like it makes me learn about some stuff that I wouldn't have learned about otherwise. And also,
yeah
I think I mean maybe we should discuss this more later too, but I think one of the things so with the mRNA people, I think one thing that I've seen discussed a lot is that like apparently one of the seminal papers that was the basis for this prize actually ended up getting desk rejected when they tried to publish it.
yeah,
Uh,
from two major journals, from Science and
yeah,
Nature, like they
yeah,
were
yeah.
rejected
Which I think is
both.
interesting because like you're talking about like oh what it shows, and I think one of the things that it shows is like how bad we are at actually judging the impact or of predicting the impact of science like when it is being submitted and published.
Yeah, like or like how important it could be to society in
Hmm.
the future, right? Like
Hmm.
one of the investigators, she had trouble like landing grants. Like she had to shut down her lab. Like she lost her lab because she couldn't like get grants to study mRNA viruses, and she had to
Hmm.
like move, like she had to move like back to Hungary. And I was just like sat there and I was just like, wait, this Nobel Prize winning scientist couldn't maintain like grant, like so like it's like, even like the best can have
Hmm.
trouble landing grants. Like it's like you're not landing grants because you're not a great scientist. It's like it's just like that's the luck
Yeah,
of the
it's just
draw.
like
You
it's a
know
thing
what I
that
mean? It makes me
happens.
feel like okay, it's
Like
okay.
it makes me feel better about the grant apply. Like I'm like, oh, actually, that one could have been completely genius, and just
Yeah.
yeah,
And you could just totally never like not be recognized. Like I was like really happy that she was recognized for
yeah, exactly.
this
So
award.
do you want to start with that story since we're talking about it?
Oh yeah, sure, sure. Like let me attempt to read their names without like butchering it. um, the award for physiology and medicine uh, went to Catalin Carico and Drew Wiseman for recognizing their work that led to the development of vaccines, and specifically the COVID vaccine, which helped us during
Mhm.
the pandemic. And
So
and I
mRNA vaccines,
yeah,
or
specifically mRNA vaccine. So
mhm.
specifically the like I guess I can feel like technology that was behind both the Moderna and the Pfizer vaccines. And I was just like, oh, like was like when I first like read the head post, and I was just like, oh, this must be like the lipid like capsule to like get it into the human body. And then when I read deeper, it wasn't that. And I was just like, oh yeah, like lipofectomy, like to transfect, like to introduce like DNA into a different system using like these like little bubbles, lipid bubbles. That's been happening for like 40 years. I was like, that's strange that that's like the reason it won. But then I read deeper, and it has to do with the base pair modifications. And so what they were doing, so 15 years ago, this was like super fringe because back when they couldn't get funding and they were still at UPenn, they were like, Drew was a immunologist and Callan was I think she was a biochemist, and so they were both trying to basically use mRNAs to create vaccines, and well, not specifically vaccines. They were just trying to like see the difference between um, uh, when you're using human like actual humans are using the mRNAs to make proteins, and when like a in vitro model, like in a dish in cells using bacteria to create the same proteins, and like what was the difference between those. Like that's like the basic question they were asking. And the difference they found was that so when you make mRNAs, oh actually maybe we start with like traditional vaccines. So
Mhm.
we don't like confuse people. So like traditional vaccines really early on. If you have questions, oh yeah, that's just you.
Oh yeah, yeah, that's
Yeah. Okay. So really on like Cliff Notes version, like the OG vaccines, like smallpox is like a weakened version of the virus, and like you just introduce it in the skin, like that version of the virus. That's like the original version of the virus. Oh, sorry, vaccine. It's like give a weakened version so your body has a chance to fight it earlier, and then when you encounter the real thing, like your body already has antibodies. So that's one version. Then came the versions that a lot of us are familiar, like flu vaccines and even things like Ebola vaccines. So those introduced like DNA of the DNA from the virus, and then it created proteins the that your body could learn to create antibodies, and then it's vaccinated. So this third version. So the problem with those vaccines is that it's really slow. It takes a long time to develop, and it involves like growing a lot of cells. So you have to like grow a lot of cells. You got to transfect these cells, and then the cells need to make the thing, and then that needs to be up. So it took a long time. So basically, it would need like 3 months. Like once you like figured out what the virus was, figured out like how like what the genetics of it was, then you had to like transfect a bunch of cells, and then like grow a lot of cells, and then like collect the material. So basically, it took a long time, and it was like hard to scale up really quickly, and so that's why the flu vaccines every year they like took time. Anyway, so that's the version that we were most familiar with, and so a lot of people on the fringe about 15 years ago were like, wouldn't it be great if we just use a single strand like of mRNA and you can like engineer it really quickly, and they were actually able to engineer it really quickly, so we can make a lot of mRNAs, and you can make a lot of mRNA and make that mRNA make protein really quickly in a dish. And we've been able to do that for ages. But the moment we tried to put it into a person, our body was just like, degrade. This is like bad stuff. Like we don't care.
And so, in theory, the reason you could use this as a vaccine then was because you can just tell your own cells to make the protein that is the thing we're trying to vaccinate against.
Yeah. So previously, we could try and like Ebola vaccine, for example, we could make it express the DNA, and then DNA needed to make the mRNA, and the mRNA needed to make the protein, and then it needs to like ramp up. And so that actually was really effective, and it did work. It's just a lot slower to design new things against new strains because you would like it just takes more time. So like the like the pro of doing an RNA vaccine is that it's single stranded, and it just literally goes straight from mRNA to protein and just makes a lot of it really quickly, and then it gets degraded and then it's gone. So like that part of the technology like was really enticing. But so the reason most like DNA is like more stable is that it's double stranded, and so like it doesn't get like cleaved by things. It like doesn't get eaten up and like just broken. RNAs are not. So mRNAs are just like single stranded, and they're like kind of flopping around. They're really easy to get cleaved, and also in large amounts of it would get like attacked by like our immune system. So basically, our immune system would create like a lot of cytokines would get in, like would ramp up, and then like it would just come and attack it and break it down. And so it would just not make a lot of protein. So once you try to transfer it from dish, like making like mRNAs making proteins in a dish to mRNAs making things in the body, the amount of protein it would actually make would be really low. And so it was really unsuccessful for many years. And that's why for like 10 years it was kind of like toiling in the background. So the reason these two scientists' work has been like highlighted for a Nobel Prize, or they're awarded this prize, is because they actually realized the difference between making it in a dish and making it in a human was that this natural phenomenon where of the uracil. So there's 4 base pairs in our mRNAs. So for example, the uracil one will get like a version that's like pseudo uracil, and this happens naturally. It just gets modified, and like just like things clam onto that mRNA, and because it's modified in the body, our body recognizes it as not a foreign substance, and it doesn't attack it. And so when you make the mRNAs in a dish, it doesn't have that pseudo uracil. It doesn't do that base modification, and without that, our body recognizes it as like an invasive thing, and then immune response, and then like it's kaput. So Yeah, so they recognized that in 2005, and this is the paper that they couldn't get published anywhere because they were like, "This is really important," and no one cared. Um, so and it eventually published in this journal called like Immune or something. Like, no, it's not fringe; it's a real journal. It's just like it's not Science and Nature, right?
Mhm.
And then like fast forward, they started a company and they've been researching this in the background for years and like just really trying to like get it to work, without like in and like proving like what the system was, like all the immunology like cascades and stuff. Um, so anyway, yeah, that's why it's so exciting. Like both of the mRNA vaccines that we recognize, at least in the States, as like the ones that like really worked and like got us, out of the pandemic. It required like using modified uracil in the like that's like one of the main components of that mRNA. The mRNAs needed to have a modified like pseudo uracil, like modified base pairs in there to protect it from us, basically from killing it, so that we can make it. So um, yeah, that's that's why they won a prize. I think it's really cool. It's really nice to have an underdog story. Like I really appreciate
Yeah.
it when it's kind of like, yeah, these people really believed in their science and they just kept at it. Like funding be damned, and then like here you go. Like I mean, I think they knew. Like a lot of people recognized that it was quite useful and it's really important for the field. Like in the past 10 years, it's just it started in a small place, which is
Yeah.
it's really cute. I like that.
Yeah.
It gives us
Also, Dutter Butter said that our stream quality is better than usual today, so that's interesting. I wonder if it's because I'm posting instead of Nadir. But also, hello.
hello, welcome
But
back to the butter.
yeah, we're back from Hades. But okay, one question I was gonna ask is, so there was a couple of people who won this one, and were they all on the same research team, or were they just people who came up with the same idea at the same time?
Yeah, no. So these Kariko and Wiseman worked together. So like they were
Okay, yeah.
both in UPenn at the same time, and like there's like a cute story about how they met at like a coffee like print at the printers and stuff. Yeah,
Sorry, was on a roll.
I was on a roll about just like fangirling about scientists. Yeah, sure. Um, yeah, no, like they knew each other. Like Wiseman, I think was a slightly more senior scientist. I think he's still at UPenn.
Mhm.
Yeah, and like it just their work just matched up. Like they had conversations, and then this thing happened. I think that's like the beauty of I mean, I don't know. It's like a really good fairy tale story. It's like, oh yeah,
Yeah.
you're just hanging out, trying to get your print. You're like making the printer work, and then like you talk to his colleague, and then you're like, hey, like I'm working on this thing, and
Yeah.
it's like
I can see, and I feel like this is more just kind of an indictment of the publishing than anything. But I can kind of see why this was considered not that high impact, though, because like when you think about it, like before like you had a apocalyptic pandemic of like a disease that required this technology, like it's like, okay, it's this very obscure thing. It's like if you substitute uridine with pseudouridine, you can prevent this particular mechanism that breaks down RNA, right?
yeah. That's true, but I think that's like the beauty of basic science. It's just like, oh yeah, like this is how it works in real life. And
Yeah.
like when they were just making this discovery, like I don't think they were out to be like, let's like make a vaccine for a virus in the future that's gonna come. Like they weren't thinking about that. They just wanted to know the difference. Do you know what I mean?
Right. They
They're
were
like,
like I can get that because I've had experiments that have felt similar where it's like at some point you just want to be like, wait, why isn't this working? Like why does it make the protein here but not here?
yeah. And like to be fair, like for decades they were injecting this into mice, and then the mice would like just get sick, and then like there would be like no protein left because like the animals would get sick because their bodies like basically creating all this inflammatory response, like the immune response to this to the RNAs existing in their body that shouldn't be there, and then they just like break it down, and then no proteins. um and also like all the other vaccines were working, right? Like I think what's really exciting about this is that these mRNA vaccines are not just going to be for COVID. Like we're going to be able to use it for all kinds of viruses that we experience that they're coming, right? Like other
Mhm.
diseases are coming, and now we have a way faster way. Like literally we can like get a virus off the street, like off whatever, like sequence it, and then like create a vaccine for
Make a
it.
vaccine against it.
Yeah,
Yeah.
like we can do it in like hours. Like that's how quick. Like we went from like something that would take months to like grow enough cells and to like create the vaccine to like I don't know if it was months. I feel like they got it down to like maybe like a month, but like to like hours. Like we're working on a different time scale now, which is like super exciting. Um, yeah. So get
So
your
you feel like
thoughts.
this one was deserved, it sounds like.
Oh yeah, I think like I didn't know who was gonna get it because I wasn't really in the field, but I think I think this is a good one. Like they could have given it to the people that put it all together, right? And then it's like it ends up being a company. I think the problem with okay, so that's the pro side of like a prize like this. The problem with it is that a lot of like current science is not done by like two fringe scientists. Like this is kind of rare. Usually it's like a team. And
Mhm.
then like ends up like only recognizing like the team group leader, or it only recognizes like like a few people, because I think you can only like max give a single prize to like three people like or maybe
Mhm.
four. Like I haven't seen them give a prize to like a group of people, um like at least not in physiology and
In
medicine.
science, yeah. Like I
Yeah,
think
in
like the
science.
European Union got a peace prize once, but
Oh yeah, that's true. Like for like climate change, I guess like, they do give it to groups, but for science I feel like they usually give
yeah,
it to
it's just it's not usually how they do it. Yeah.
yeah, two or three people, and like recognizes. I mean, like the thing is like the reason we have these vaccines is not just because of these two people, right? Like these two people were really important, but like it is the work of many scientists and like years like just toiling away. So like on one hand I'm like I'm really glad they got it. On the other hand it's like, oh hey, this prize, like does it's not every it doesn't get every it doesn't include all the humans that worked on it, and there's a
Mhm.
lot of really amazing humans all over the world that were working on it. So um yeah, I don't know, it was fun, I
Yeah,
think.
but it was good. Yeah.
Yeah, like I wouldn't put like too much stock on like who wins the prize and like are they important to society. But it's nice, it's like a fun activity to see. It's kind of like who won the Oscars this year. It doesn't make them like the best actor in the
The
world.
best scientist. Yeah.
Yeah, you know what I mean? Like it's just like yeah, like like of the movies this year, like that one had you had a great performance, and I think these two humans for their scientific careers had a great performance, so they deserve the
Yeah.
prize.
I was gonna say that reminds me of some of the other Nobel prizes, which I was reading about them, and I like it. I mean, in part because like I'm not in these fields, but I'm like, oh wow, this is like not necessarily a thing as I would say is like one of the most important scientific things ever, but it definitely is really cool to read about. I think
Yeah.
one was the one in physics, which was for basically for attosecond pulses of light. Um, so an attosecond is a very just like it's an incredibly short period of time basically. And so the idea with this was that if you can generate pulses of light that last just an attosecond, you can image things that are happening really fast. So the idea is that if you have a camera and you illuminate something with a pulse that only lasts an attosecond, it basically acts like having a very fast shutter speed, and you can capture very fast things without them blurring, right?
I was gonna say, is it kind of like 2P imaging where you just like let a couple photons come out and like bounce back?
I'm not familiar with P imaging, but like the
Like the idea is that like
Mhm.
it's like such small amounts of light, like
like
that they
just two
have.
photons.
Yeah, yeah, yeah. Like I'm like that's kind of like 2P imaging. Like
Yeah, I think
it
so.
requires a very small amount of light.
I mean, I think in this case, like it can even be an intense light, but the idea is that it's a brief light. So kind of the idea is it's similar to like how a strobe light lets you see things that are happening very fast. So kind of like if you have like a hummingbird flapping its wings, it just looks like a blur. But if you illuminate it with a strobe light, then you actually see kind of the wings frozen at one position if you match the frequency right. And the reason is because kind of when the strobe light is off, just there isn't enough light reaching your retina to influence the image. So there's just kind of nothing there. And because the image can only be formed when the light is on, and the light is on for such a brief period of time that you're really getting not that much blurring. So that's the underlying principle of why we use these kind of very short pulses to image fast things, is that like it's essentially just it lets you can have a sensor that responds fairly slowly, but if you're only illuminating it with this very fast pulse of light, you can image things clearly that are moving very quickly.
Got it.
And
So
so
what are they using it to image?
so electrons. So
Oh,
so that is apparently the big like thing that you can use this for, is that you can actually use it to take pictures using, and when I say light, I'm actually talking about light that's at very high frequency radiation, because visible light actually is like too low a frequency to be able to see electrons, but you can use higher frequency light that works. And essentially what this lets you do is it like lets you take a snapshot of just where are the electrons in the atom at this moment.
oh, that is actually kind of cool. Very cool. And so, like, like, are all our models correct? Like, it actually, it's just
It
whizzing
seems to
around
generally be. Yeah, I mean,
in
I
these
mean, so
clouds.
it's tricky because like the electrons also have weird quantum phenomena, which is what the other Nobel Prize is about, actually. But um,
Oh,
anyway,
because like imaging it will like disturb it,
right?
right?
Yeah. So you can get an image and you can see like where were the electrons when you observed it, and I think the theoretical model of how electrons work is a lot weirder than that. Like they don't really necessarily have discrete positions when you're not looking at them, but they do. But then when you do look at them, there's kind of a statistical probability that they're more likely to be found in certain. It's quite weird. But the main thing is like this is actually very valuable for studying certain kinds of chemical reactions, and it's generally allowed us to figure out what is actually happening in certain kinds of chemical reactions at the level of electrons. Because what you can do is like you can have a theory of kind of like when these two molecules are interacting, the electrons are interacting in these particular ways, and now we can actually look at it and be like, are the electrons actually doing that or not? Um,
Oh, that's so cool. Like you don't have
yeah.
to wait for the reaction to finish and then like figure out what
Right,
happens
and
in between.
it tells you why, because it's like okay, you put these two molecules together and then you get this other molecule, and you have a theory about all the steps that are happening, but kind of this lets you actually see why. Or if you have
Like
materials.
you can monitor it like
Right,
while the reaction's happening. Oh, that's very cool.
right,
Okay.
and it's like and it lets you kind of validate the theory. Or you might have materials with really weird properties, and it lets you see like what is actually making the properties of this so weird. Or you have things that emit light in weird ways, and you can see how the interactions with the electrons are actually allowing it to do that. So
Okay.
it's
That's
used
really cool.
it's used to kind of answer a lot of the like why and how questions, I think, is what I was getting at.
Oh,
But
that's really cool.
I wanted so I was oh sorry, were you gonna say something? Yeah.
I was just gonna say, like, it almost feels like, why is this a chemistry prize? And I understand why it's a chemistry prize now, but it could have easily been a physics prize. Do you know what I mean? Like,
Uh-huh. Oh, this was physics, I think.
oh,
Yeah.
sorry, this is the physics prize. I was just like, wait, is this the
Yeah,
chemistry prize
physics.
or the physics
The
prize?
chemistry one is also very physicsy this year, which is why it's confusing. But yeah, I was all so the other thing that I wanted to point out is that it's actually like so I was reading about this, and it's a lot more intuitive than actually I thought it was going to be. Let me pull up my screen. Um, and also, so the other thing that I learned is that the Nobel Prize website actually has some really good summaries about like
Oh,
the
Drost.
science, the science behind all of these. But anyway, the way that this works is it's based on this observation um, this observation that when you shine lights through certain kinds of noble gases, the gases actually generate overtones. And so an overtone is essentially just a kind of fluorescence where the gas is emitting the light that you shine, like the light that you shine in is passing through the gas, but some of the atoms are absorbing that light and emitting light at a higher frequency. And so we call that an overtone. And essentially,
Mm.
um, and so what? Oh, they don't have the list of, but yeah. So there's three people on this Nobel Prize, and so what this first person and Lehuelier figured out is they were the first one to show that this effect happened. Um, but the cool thing about this is that so when you shine a light through gas at the right frequency, you get kind of these different frequencies of waves that the gas is emitting. And those waves then kind of interfere with each other. So, because all these different frequencies are present, what's happening is that the peaks and troughs kind of
You
overlap with each
and
other.
like they combine and you
Yeah,
create the
exactly.
bigger
And
wave.
so
Yeah.
what you get is this step where it says they reinforce or cancel each other out. Um, and what that means is that instead of this continuous light, you essentially get these bursts of light coming out of the thing. And this is happening in the ultraviolet range when they first discovered it. Um, and the cool thing about that is that you can tune that so that these bursts of light are very, very short, like on the order of attoseconds. And then just by shining light at the right frequency through the right kind of gas, you can then generate these extremely brief pulses of light, and that's how it works.
That is actually really cool.
Mm-hmm.
Oh, that's a cool one actually. This one's like, yeah, that's cool. Now I really want to read into it more deeply now.
Yeah, and like there's some more complications to it. So like one thing is, so one of these people, I think it was yeah, let me, oh yeah, they say, oh yeah, so Lehuelier discovered this overtone effect. Pierce Agostini was the first person to use it to generate these very brief pulses of light, and then this third person, Ferenc Krauts, um, came up with a different protocol that used the same effect, but it was able to produce just one pulse of light, and then it stops, which is also obviously useful for something to only image.
Yeah. So wait, these three scientists do not work together. They're just
They
like,
don't know. Yeah,
oh,
yeah,
okay.
yeah.
So
So
they were just like simultaneously working on really similar things in different
mhm,
ways all over the world. That's
mhm.
also a really cool story. Oh,
Yeah.
Frost.
Yep. Um.
That's so cool.
Anyway, yeah. So that's the attosecond pulses story. And then should I talk about the chemistry one, which is also
Yeah,
very,
yeah. Do tell about the
yeah,
chemistry one. That one felt like a chemistry one. I genuinely thought that was a chemistry prize because you're like, yeah,
yeah.
you can
So
look at reactions happen in real time, and I was like, if I was a chemist, that would have been awesome. What is
yeah,
the
I
chemistry
don't.
one?
Yeah, I feel like the chemistry one, like I said, was also very physicsy. But hang on, actually, let me pull it up. So it is about quantum dots, and it is again three kind of unrelated people who all just happened to be working in this field. And so first of all, so the first thing that I was trying to figure out is like what is a quantum dot, and it is quite confusing. Um, but basically these are very small pieces of semiconductors, so it'll be like basically a silicon crystal that consists of just a few thousand atoms or so. And so the interesting thing about that is that these have this property that they say, I'm trying to find where they call it. What was that?
I didn't say anything. I was just like,
Yeah.
tiny speck of matter.
Yeah. So the interesting thing about this is that because of quantum phenomena, when you make the crystals that are this size, they essentially act like they're just one atom. Um, and lets them have a lot of really weird properties, but one of them is that you can make them fluorescent and you can tune what wavelength they fluoresce at by changing the size of the quantum dot. Um,
It's I'm still stuck on the fact that it's a zero dimensional
yeah,
thing.
I don't know what that means, but
It's a single point. It's like not
I
even
guess
like
yeah. So
so
I
like
guess
a
it's
piece
just
like a dot on a piece of paper is one
yeah.
dimensional. So like this one is like not even on a piece of paper.
Yeah, I mean, wait, I thought a dot, but a dot is like a dot is zero dimensional, right? And then a line has one dimension.
And line is one dimension. You're right. You're right. Sorry.
Yeah.
Yeah. It's just a dot. Yeah. It's
Yeah, so I kind of get what they're saying then. It's saying like basically it acts like it doesn't really have an internal structure. Like it's like it kind of it has a bunch of electrons in it, but they're all acting like they're just part of one atom.
yeah.
Um anyway, yeah, so the basic principle of this is that so different kinds of atoms will like fluoresce at different colors, and you can see this like when you burn different substances, they'll make different colors when they're burned. And basically that has to do with the layout of the atom and how many electrons it has in its outer shell. And the colors that it emits is basically determined by electrons jumping from kind of their lower energy inner orbitals to their higher energy outer orbitals. So, and the interesting thing about this is and this like so that really determines like a lot of the properties of materials is how the electrons are arranged and how many are in their outer orbital. Um, but the thing is we don't know how like we can't really manufacture custom atoms that have the properties we want, but what we can do is we can make these chunks of semiconductors that have the properties that we want because they act like they're just a single atom. And so that's really the kind of the key thing about this is that um, they they like so these chunks of semiconductor show the same behavior as an individual atom would, and then so depending on their size you can make them behave in different ways and emit different colors of light.
Got it. Oh, this is so okay. I've always found it really weird when a particle just switches from like Newtonian mechanics to quantum mechanics. like
Yeah,
what about its size do you just like flip the switch? Do you know what I mean?
yeah.
Like at what size? I mean, there probably is an answer to this. I'm just not a physicist. I
So
have no
actually
idea.
no one really knows.
Oh, okay.
um
So no one knows.
yeah.
Like why does some things follow quantum mechanic rules? Like,
Mhm.
and then why like what makes the other things follow just like the Newtonian mechanics? Do you know what I mean? Like
Yeah.
what makes where does it make that flip and why? Like, because some of the particles, I don't know what size they
Mhm.
are, but like they're like, yeah, all these particles follow these rules except for these three, and I'm like, wait, why?
Yeah. Yeah, this is apparently one of, and if folks in the chat have like read more about this, like feel free to chime in because my knowledge about this is very cursory. But like, this is one of the like less well understood aspects of quantum mechanics is like why what causes that to happen. One theory is that like it actually doesn't happen. Like, so the idea is that just in large scale things just there's still quantum stuff happening, but just kind of because you have such a large number of particles, just you're seeing kind of the average of what's going on, which is obeying the classical mechanical rules. But also, people don't like some physicists don't really like that because it seems like there are some experiments where things make very sharp transitions between acting like in a quantum way and in a classical way, and often like so quantum physicists will talk about observation a lot, like kind of like a system works in the quantum way until you look at it, and then we're still not exactly sure why looking at it causes it to change like what it's doing. I
Yeah.
mean, I think the conventional way that like physicists will talk about it is that when you're observing it, what's happening is that this very simple isolated system is interacting with a much larger, more complex system. It's really this that kind of interaction is what causes it to go into the classical mode, but it's quite mysterious.
Yeah, this
Um,
is mysterious. We need more physicist friends. They
anyway,
would
um,
jazz up our conversations. Very cool.
yeah, but anyway, that is one of like so that's also related to why these act like individual atoms. Is that for some reason that we don't completely understand, if you have this just chunk of homogeneous silicon surrounded by just kind of medium or water or whatever, um, it stays in a quantum state.
Got it. Did they show us like what kind of applications or what kind of
Yeah.
experiments? Yeah, like they is it in the on the website?
Oh yeah, so they mentioned a couple things. So one thing is that these are actually used in displays a lot, um, and so the biggest application is that you can change the size, or you can get these to emit light, and the frequency they emit is like continuously tunable. Um,
Oh,
so they're being used in pretty much every application where we need colored light. The other cool thing about them is that even if you have one of a certain size, often by changing some other properties like the voltage that you apply to it, you can also change what frequency it's emitting. So these are used in displays to just produce different colors of light.
you mean like monitors, like or screen
Yep. Yeah,
monitors and
yeah.
everything?
So a lot
Okay.
of monitors that you can buy right now are quantum dot displays, and they use these.
Oh, that's really cool.
Uh-huh.
Like I didn't realize like how widespreadly used like
Mhm.
this discovery was.
Yeah, yeah,
Very
yeah.
cool. Yeah,
And then the other
world's
big
connected.
one is kind of like dyes and markers, interestingly. So I think one of the interesting things about these is that so like I mean, and you know this, right? So most chemical dyes will eventually bleach. Like after they've been exposed to enough light, they just kind of stop working. And it's because they're doing sort of an internal chemical process that depends on the structure of the dye. And after it's been exposed to enough light, that structure breaks down and it stops working. But these quantum dots don't really have an internal structure. They're zero dimensional, right? So they don't do that. And they are extremely durable and will basically continue fluorescing as long as they're around. So one of the one of the things that they're actually being used for is like labeling, like kind of biological tissue labeling. So
Oh,
you can attach these to reporters that will bind to specific components like in a cell and make them glow. And they're very versatile for that, because you can get like a ton of colors of them just by changing the size of the quantum dot. And they seem to be pretty non-toxic, and they don't give out even after you've been imaging them for
oh,
a
so
while.
like they don't bleach.
Yeah.
That's awesome. Like imagine
Uh-huh.
like a fluorescent protein that doesn't bleach.
Mhm.
That'll solve a
Yeah.
lot of problems
Yep.
in biological sciences.
So that's another application that was mentioned. But
Oh, that's really cool.
yeah.
Cool. Yeah. So that's a. I feel like we came back strong
Uh-huh.
after
Yeah. Yeah.
hiatus.
Yeah. Uh-huh.
We're like here's some like really cool science.
Yeah, like that's how I felt about like I was like that's what I was saying. It's like, oh, I feel like this made me the Nobel prizes like turned me on to some cool stuff.
Yeah, like it just made you like kind of read a little bit something peripheral to
Mhm.
what you would
Yeah.
normally read. Um, I since I just came back from vacation, I thought I would share a couple photos
Hmm.
from my trip. So I went to Stockholm as one of my stops on my bop around the world after my PhD. And I went to the Nobel Prize Museum. It's a very small museum. I have to say the website, the Nobel Prize website, is probably as good as the museum, unless
As in,
like yeah, unless
mm-hmm.
you like personally know someone that won one and like want to go look for a specific thing. Um, but there were several things that was really cool. Let me share my screen. How do I share my screen? I know how to share my screen. Share my screen window. I think there's nothing weird on my screen. Yeah. Okay, go computer go. Um, is it showing the screen? Is it just Drost affecting?
It is. Hang on, I have to like put the screen on. I think there we go. Yeah.
Uh,
Okay,
are
now.
you seeing are you
Yeah.
seeing my reels?
I'm seeing your Stockholm photos.
Oh,
Yeah.
okay, you are. Okay, cool. Okay, so this is a Stockholm photo. Here is like in the museum. Okay, so I think this is the entrance of the museum right here. And when you go in, there's like literally a giant medal on the ground. I thought I would show a couple of things. So when you win the Nobel Prize, they ask you to donate. Oh wait, here's a picture of me with one of my
Winning the
favorite.
Nobel Prize. Oh.
No, this is one of my favorite humans that have won the
Mhm.
prize. Um, but I thought this is I took a picture of a couple of the artifacts that I thought were really cool. So sorry, this one. Uh, Here, I think this is a good one. So this one was from the scientist Barry Marshall, who proved that ulcers were caused by bacteria, and this is the vial where he grew that bacteria and drank from and gave himself ulcers.
You feel like he got a Nobel Prize just because like they felt kind of bad for him, like.
And this is his chair. I feel like yeah, maybe they just felt bad for him. I thought it was really cool. It was a cool museum. Here are some of the things that I thought was really cool. There's someone's notebook, and this is like in their lab notebook, and like they wrote like "Holy shit" in their
What
notebook.
was that in reference to?
This is I think this is a black hole observation
Oh.
from a couple years ago. Here's the full notebook. This is uh Joel Adlock and Wayne Black. Yeah. Like I was just like, I was just looking at their notebooks and I was like, yeah, this looks like one of our notebooks. Like beautiful, slow pattern. And then this one, holy shit.
Holy shit.
Yeah, like she just like submitted that page of her lab notebook into it. This person,
So you were saying they ask you to donate something to the museum, or?
yeah, they asked them to donate something to the museum, and then like that's what they kept there. And then this one was really cool. So these are actually no Alfred Nobel's like personal effects. Like he traveled a lot too. This is his like travel bag, and like this is what he had in it. And I was like, all he had was books. But you
Is
can see
this
it's like
like, did it make you feel better about like transporting so many books?
I know it did make me feel a little better about transporting like all these books in my house. Anyway, I thought I would share that since
Nice.
that was Nobel Prize adjacent. Um,
Mhm.
yeah, no, it was really fun. It was cool to just see a museum. I think
Yeah.
the websites are way cooler. Actually, I think they spent a lot of time making that website quite good because it's like targeted towards a lay audience. And like you'll go to the museum and there's like all these kiosks, and then you just like click through it, and it's like the website on a touch
Oh, and
screen.
they just have the website up in the museum.
Yeah.
Yeah.
Um so besides like those like couple exhibits where like they have like these artifacts, the rest of the museum was just like these kiosks where they explain like how they choose the nominees and like why these people won. And it was cool. That I thought I would share.
Nice. Yeah, that was a cool addendum to the Nobel Prize stream. Yeah.
Yeah. Anyway, I think we rolled through that a lot
Yeah.
faster than I thought
Yeah.
we
That
could.
was fun. Yeah.
Yeah, that was a lot
Oh
of fun.
yeah, and yeah, just I've also like if you're curious about like we mentioned the Nobel Prize website actually has some really good like lay explanations of all the stuff on it. So
Yeah,
yeah.
I sometimes go into history of science like for fun. Like I like to look at like people's lives sometimes because I'm like I don't know if it's like I read like memoirs and like autobiographies or other people written biographies. So sometimes I'll just like get into like a rabbit hole and I'll just like read up on certain scientists, and I find that's actually quite fascinating. And it's like fun to see like what their lives were at, or like at some point they got scooped and then like or like someone else got scooped and then they kept going. And there's like a lot of fun stories. Like I wish they were. I don't know. Maybe they someday they will be like movies, and some of them have become movies, for like lay audience. It's actually quite dramatic. A lot of their lives are really cool and interesting, and it's just It's very rare that scientists' lives are on the main screen, so it's nice to
It's
see it.
cool. Yeah.
Anyway,
Yeah. So should we sign off till, and then we'll
yeah,
be
let's
back
sign off.
next week.
Thanks.
Yeah.
Yeah, thanks for joining us. Next week, I guess we're making pinhole cameras, and we'll talk about some cool things in the sky. Yeah, look out for auroras,
Should
I
I talk
guess.
about the UFO project? No.
Yeah, and we can talk about UFOs. Every time we post about UFOs, we get a larger audience.
Yeah, so we should, but I don't know. We'll see how it goes. We might talk about UFOs, but yeah. Anyway,
Very
night.
good. Thank you. Nice to see you. We're Philosophising.
Yeah.
Bye.
Bye.